several reasons. One of them is that I can speak Hebrew with him. That's very important. But Lior has been a friend for a long time now, and we both graduated from the same institution, which is the Technion, the Israeli Institute of Technology. Where Lior received his MD and PhD, PhD in biophysics and physiology, which is part of the medical school. I think at the same time that he worked on the PhD, we were also working with the Biosense toy to develop methods for three-dimensional mapping, and electroanatomical mapping, they called it. That's what it was called. And so Lior was instrumental doing experiments in pigs, if I remember correctly, on 3D mapping using the Cardiomyocyte system that was developed then. So you see, that's one side of Lior, which is the clinical side. And he's still a practicing cardiologist who does EP service at the Rambam Hospital, which is the academic hospital for the Technion. The other hat that he wears is stem cells, and he's done a lot of work on stem cells in many different contexts, but what is really very interesting to us here is the context of cardiac arrhythmias, and especially the hereditary cardiac arrhythmias. He's used stem cells as a model to try and model those disease processes. The other thing that I wanted to say is thank you all for being here. I know that this is not an optimal week because many people have gone home and many people are with their families. It's Thanksgiving week and holiday week. So whoever is here, have a great and happy Thanksgiving. So here.
Thanks, Yoram. It's actually a lot of fun. It's my first time here, strangely enough, but it's been a lot of fun, and I've been enjoying and learning a lot, and I'd like to thank you for the hospitality. What I'll do today is talk about a little bit about the work that we are doing in the lab involving human pluripotent stem cells. We have other interests in the lab, but our main focus on human pluripotent stem cells. And since this is the interest of the group here is focused on electrophysiology, I'll try to discuss some of the applications of these cells for electrophysiology. And if we can think about it, there are several electrophysiological applications to cell therapy in stem cells in the heart. We can use this model to study developmental electrophysiology, how excitability and calcium handle developing in early cells. Since this allows us to generate human tissue, we can use these human heart cells for drug screening, drug discovery. We'll talk about the potential use of these cells for modeling of inherited and acquired disorders, but also for the field of regenerative medicine, which I'll talk about. If you want to use heart cells derived from human stem cells to repair damaged heart, there are several electrophysiological implications. For example, if you want the cells to contribute directly to mechanic contraction of the heart, then you want them to integrate electrically. One of the potential side effects that we are really worried about is the issues of arrhythmias. One could also think of using cells to treat arrhythmia disorders. And we have a number of projects for the latter, but I'm not sure that we'll have time to talk about. So the first stem cells that we worked with was human embryonic stem cells. These cells can be derived from the inner cell mass of human blastocysts. So this is the last time where cells still have the capacity to generate any cell type in the body. So in 1999, 2000, we worked with Jamie Thompson in collaboration with Professor Itzkovich from our group, from our hospital, who's the head of our OBG. They were able to establish the first human embryonic stem cell lines, in which these cells can be propagated in the undifferentiated state, and then due to their embryonic origin can be differentiated to any cell type in the body. So a little bit more than 10 years ago, we were able to generate the first cardiomyocyte differentiating system from these human cells. The original system was rather spontaneous differentiation. We grow the cells in the undifferentiated state. Then we transfer them to suspension, where they form these three-dimensional cell aggregates called embryonic bodies. And trying to stimulate the natural process of development, after a week we plate them on culture plate, and when you optimize the conditions, you can add growth factors and so on. We were very happy to try to see the first beating heart cells in the dish. So we were excited because this was the first time that human heart cells were generated in a dish. And these cells beat forever. I've been sewing this slide for more than 10 years and they still beat in the slide. But the longest we've held them is more than a year in the dish. We then wanted to ask whether these cells are limited to isolated cells or whether they generate a functional syncytium. So to this end, we adopted the microelectrode array mapping technique that was originally developed to map electrical activity in neuronal cultures. So we adapted them to the cardiac tissue, cardiac cultures. One can record extracellular electrograms from these tissue. You can, for example, plate this beating area on top of this chip, record extracellular potentials, and then analyze them the same way you analyze electrograms in the clinic. So you can determine, for example, the timing of the electrical event at each electrode, color code this, and get these activation maps that show conduction within this tissue. So this indeed shows us that this differentiation system is not limited to generation of isolated cells, but rather there's a functional system with conduction between the cells. So moving forward, the next important question that we ask, if you want to use these cells for regeneration, it's not enough that you generate heart cells or functional tissue. You would want the cells to integrate electrically with the existing heart tissue if you would want to improve cardiac function by contribution to contractility. So the issue of integration into existing network, we believe, is a very important issue in all forms of regenerative medicine in the body. So we started with an in vitro study in which we took our human heart cells, as you can see here, put them next to rat heart cells, and examined the co-cultures, and as you can see, within a few hours, the two tissues started to beat in synchrony. So when the human cells, this cluster beats, also the rat beats. So to determine whether this is true electrical integration, we performed this study on top of a microelectrode array on the MIA plates. In this example, the white area is the human tissue here. The rest is rat tissue. Note one electrode, the red electrode from the human tissue, and one electrode, the green electrode from the rat tissue. And you can see that there's one-to-one conduction between the two tissue types. If you pace one of the electrodes very fast, there's still one-to-one conduction, either when you pace the rat tissue or when you pace the human tissue. If you look at one beat and generate a map, in this case, the activation originated in the rat area and then propagated and activated also the human tissue. So at least in the dish, there's integration, the cells form gap junctions, and there is action potential propagation between the two tissue types. Will that happen in vivo? So it's not as easy to answer that. We did a few studies. I'll show you one or two examples, and other groups have added to that. So in this study, what we did, we labeled the cells with a fluorescent marker. We did this together with Jeff Olgin at UCSF. We then injected the cells into the rat heart. And 2 weeks later, we used the Langendorf apparatus and optical mapping system to study the electrophysiological properties of the transplanted heart. You can see in this case that we are pacing from this area, from these electrodes, and we are looking at conduction in this dynamic display. This is the site of cell transplantation, and you can see in the map and also in the isochronal map generated that in general, in most of the hearts, we didn't see significant conduction abnormalities at the site of cell transplantation when we transplant the human cells into the rat heart in a normal environment. In contrast, at the time it was popular, people thought that perhaps we should, skeletal myoblasts can be a source for myocardial regeneration. Okay, people were saying the heart is a muscle, the skeletal muscle is a muscle, maybe we can use cells derived from the same patient. They are stem cells within this skeletal muscle called satellite cells that one could identify during biopsy, harvest, and propagate to generate hundreds of millions of myoblasts, and then transplant back to the same patient. And this idea even reached clinical trials. But as you know, skeletal muscles have completely different physiology from the heart cells, okay? They are activated by neuromuscular junctions, they don't have gap junctions, and so on. So when we do the same experiment, taking rat skeletal muscle cells, injecting them into the heart, and then mapping this heart, you can see that in this case, the activation goes here, replacing from here in this example. The activation propagates around the area of cell transplantation with conduction delay at such site. You can see it also in this map. It goes from red to yellow to green, and then goes around this anatomical obstacle of the cells. And this can be even dangerous because in about half of these animals, just by pacing them, we could generate a reentry circuit, as you can see here. You can see a reentry circuit propagating around the area of cell transplantation, and this is in healthy, not infarcted animals. So the issue integration has important meanings in terms of functional contribution, the mechanism, and also has significant issues with regarding to adverse events, such in this case, reentry ventricular arrhythmias. And indeed, these skeletal muscle transplantation trials were terminated prematurely, mostly because of lack of efficacy, but also there were reports of ventricular tachycardias in these patients. So this is an important issue, integration. We then went to perform 2 proof of concept studies to try to see whether these cells can help an animal model of cardiac disease. The first one we described was in a model of slow heart rate. We saw that the stem cells have pacemaker cells within them, okay? So we thought, why not try to use these pacemaker cells to pace the heart in a model of slow heart rates, such as an alternative, for example, for an electronic pacemaker. So to this end, we created a pig model of slow heart rate by ablating the AV node. So you can see the development of complete AV block in the pigs with the P wave marching through the QRS complex. The pigs had a slow junctional escape rhythm, as you can see here. We then transplanted spontaneously beating human heart cells into the ventricular area, into the lateral wall of the ventricle, into the posterolateral wall of the ventricle, to try to generate an alternative focus so we can identify it. And you can see in some of these pigs, we could see the development of a new pacemaker rhythm, which was faster and had a different QRS morphology than the nodal escape rhythm and junctional escape rhythm. We could map the source of this new mapping system and this new pacing activity using the Carto system. As you heard from Yoram, we were involved in the development of the Carto system. We can then identify the source of this mapping and pacing activity and pinpoint it to the site of cell transplantation here, and we could identify the cells during histology at that site. More importantly, because most of our efforts are going towards infarct repair, we performed a proof of concept study in which we injected our cells into the infarct area in rats. We created an animal model of myocardial infarction. A week later, by ligating the left anterior descending coronary artery, a week later, we implanted our cells into the infarcted area and followed the animals by echocardiography. So here you see one parameter, fractional shortening. A normal fractional shortening in rat is about 40%. When you create an infarct, it immediately drops to about 20%. And then if you follow this model, for example, just by injecting cell line into this area, into the infarcted area, you can see that there is continuous deterioration in myocardial performance over time until 60 days. We see this also in patients. This is called abnormal remodeling. Interestingly, if you inject cells that are not heart cells, you see the same remodeling in these animals. But if we inject our human heart cells, you can see that this remodeling is altered. This is the green plot. And there's even some improvement in function over time. So we were happy to see this because this would be meaningful if we have it in patients. But nevertheless, it also showed us that we have still a lot of work to do. First of all, you could see that we are still far from regenerating the heart in a way that will result in its original fractional shortening. And similarly, when we look at histology, we see the cells with an infarct, but we see that more than 90% of the cells are lost. So only about 10% of the cells that we transplanted survive long-term. Moreover, most of the cells do not form this nice, highly organized muscle structure with the cells oriented in the same areas and mature and so on. They remain rather small and are oriented in different directions. So we still have a lot of work to do to optimize and get better results. Interestingly, there are now studies emerging in large, yes?
Do you just inject them as solubilized cells?
Yeah.
Dispersed cells?
Yeah, what we learned is that they survive the best when you inject them as small clusters of cells, not as individual cells, but 10, 20 cells together. They need attachment from other cells. As you will see, they need attachment to extracellular matrix, they need vascularization, there's other stuff that will help their survival. And there's a lot of reason why they are not surviving. First of all, acutely they're just washed away. So you need to find a way to entrap them. Then you have anoikis, which is kind of death when the cells don't have supporting extracellular matrix that they feel. There's the issue, if they don't contract, they also may undergo apoptosis. And you implant them into a harsh environment where cells die there. It's an infarct area. So there are multiple ways why they die. Yes?
Is there any difference in the type of cells that you injected here versus the ones that pace the ventricle? And how can you
how you can prevent them to pace?
Right.
So first of all, there are now ways to eliminate the cells with the more pacemaking properties. We never saw the cells pace animals that did not have slow heart rate. Okay, so this in rat, of course, there was not a problem because it beats like 300 beats per minute, and the cells never did that. But also when you do it in pigs, if they have normal heart rate, we didn't see ectopic activity due to automaticity of the cells. But this is basically many more cells, and that were not in the pig study. We tried to implant them as chunks of tissue because we figured that the individual cells would have less chance of pacing the heart because of source sync mismatches. So we try to generate a tissue that will generate action potential between the tissue and then propagate it to its neighboring. When we do this as more or less small, very small clusters of cells, we don't see that. But one of the issues is, you're right, if you want to regenerate the infarct, maybe you need to get rid of any surviving remaining pacemaker cells. So there's an issue of, so at that stage there are many challenges before one can think about using this in patients. First of all, the original differentiation system that we showed you, using this embryoid body system, results in rather low percentage of the cells becoming heart cells. That was our original strategy. There's an issue of scaling up, because in the adult human heart there are 4 billion cells. When you develop heart failure, you usually lose about a quarter of them, so you need to regenerate 1 billion cells. Imagine that you lose many of them, so imagine the number of cells you need. So scaling up is a big issue. There is issue of immune rejection, because in these animals, for example, this is xenotransplantation, human cells into animals. We gave immune suppression to prevent it. We gave drugs that prevent immune rejection. But also in human, although these cells are embryonic, they are not organs that are highly immunogenic, they are still expected to be rejected if you do not give immune suppression to the patient. We talked about integration, we talked about donor cell loss and structural and functional maturation. So with regards to generating enough cells and generating purified population, there's been a big advancement in this field. So by a number of groups that use this model to provide us signals about early cardiac differentiation, there are now a number of protocols. This is one of them that we're using, that you can get almost 80–90% pure cardiomyocytes. So the entire cells, the entire population of cells, when you optimize this, can become cardiac cells. So the percentage of cells getting enough cells that are cardiomyocytes will probably not be a challenge. I just came back from a meeting in Toronto where they have this nice bioreactor control environment where they can generate enough cells. So this is more and more being solved, getting enough cells and getting enough purified cells. The issue of survival, we are more and more moving forward towards combining cells with other approaches. And we've done significant work in the issue of tissue engineering. The idea is that our organs are not made only from cells, okay? They are made from cells that grow together with extracellular matrix that provides a scaffold for the cells, that supports the cells, and gives the structure of the organ. So tissue engineering combines cells with polymers, either synthetic or biological, that will provide a scaffold for the cells to grow. So we've been collaborating with a number of groups. This is a collaboration with a German group. I'd like to show this, although this is rather old. This is their work with rat heart cells, in which they take neonatal rat heart cells, mix it with collagen and other extracellular matrix. They put it in the mold until it becomes hard. And then we put it on a stretcher device. It's like lifting weight for the cells. So they work against the load for a couple of weeks. And then afterwards, the entire tissue starts to beat. And as you can see, you can get this spontaneously beating rubber band-like structures that can generate a significant amount of force. So together with the Zimmermann group at Göttingen, we've been trying to optimize this technique for human cells. And after many years, you can get this beating calamari looking like structures that can, you can use it, actually it's a very nice model also for human physiology and for modeling mechanical performance, because one can measure development of force and so on. And this mechanical stretch is very important. The cells align along the strain patterns, they hypertrophy and mature during this process. Okay, one of the problems of tissue engineering is the issue of diffusion and vascularization, because you are really limited in the size of the structure muscle that you can create when you transplant it in vivo because of lack of vascularization. So until the tissue creates its own vascularization, the cells in the center may suffer from hypoxia, ischemia, and undergo necrosis. As you know, the heart is especially sensitive to ischemia. In the heart, every cell has at least one capillary adjacent to it. So this could be a problem for survival of these grafts. So together with Shulamit Levenberg from our biomedical engineering and faculty, we thought of perhaps generating engineered tissue that will already have vascular structures within them. So the idea is to take cardiomyocytes and to mix them with vascular precursor cells to make an engineered tissue that will have blood vessels. So when you take only heart cells and grow them on a scaffold, in this case it was a PLLA, PLGA scaffold, you can see that we can get some brown cells. This is staining for troponin I, but not staining for CD31, which is a marker of endothelial cells. When you take cardiomyocytes and endothelial cells, you get many heart cells. You get some endothelial cells, you can see the brown staining, but not many, and nothing that look like blood vessels. So we were a bit disappointed for that. So we added a third type of cell, embryonic fibroblasts, and in this tri-culture, we could get cardiac tissue, as well as vessel-like structures, lumens, that are aligned by endothelial cells. So it's important to add a three component. These are embryonic fibroblasts in our case, but since then the field has moved after this, our initial study. Other people have recapitulated it and demonstrated that you can do the same thing with smooth muscle cells, with pericytes and so on. So we believe that the fibroblasts become smooth muscle cells or pericytes and support these vessels that are formed. We then wanted to see whether these cells, with this engineered tissue, whether it can integrate after in vivo transplantation, whether the blood vessels can become functional and support the engineered tissue that's transplanted. So to this end, we transplanted the engineered muscle that we created on top of the rat heart. As you can see, this is the in its pathology. This is the left ventricle, right ventricle, and the engineered tissue. This is a high magnification of this, so you can see the remaining scaffold material, which is degradable. It will, sorry about that. will eventually be, what do I do? It will eventually be degraded. And you can see when we stain for troponin I, you can see that this is the rat tissue, this is the human tissue, so that it seems like the tissue survives rather well, as you can see here. This is something that we have to deal with. Occasionally we see that, so there is a bit of fibrotic encapsulation of the engineered tissue. Maybe this is technical how we suture it in the muscle, that we may hinder the electrical integration between the two tissue type, between the rat and the transplanted human. And we can get nice cardiomyocytes, as you can see here, with nice striations in this engineered tissue transplanted. Now the question is whether the vessels that we created, will they integrate with host vasculature and perfuse the tissue? So initially we were very happy to see that many of this is engineered tissue, this is rat tissue, that we can see many blood vessels present within the engineered tissue, transplanted tissue. When we use antibodies targeting rat heart cells versus antibodies targeting human cells, you can see that about half are human cells and about half are rat cells. So half are the blood vessels that came with the tissue, half are rat vasculature that infiltrated the tissue. We were happy to see in many of the human tissue the development of the presence of red blood cells, meaning that the human blood vessels start to perfuse the tissue. And we can quantify this by injecting into the heart before sacrificing the fluorescent microspheres that flow with the coronary flow and become entrapped in small capillaries. So you can see that they are all over the heart and the engineered tissue, and you can see them within the human vessels. So this could be a way to improve the survival of the engineered tissue by putting the heart cells together with vascular tissue. One of the problems with what I've shown you so far is that you have to suture the tissue on top of the heart, okay? On either on the infarct or maybe replacing the infarct, which is not sure that mechanically it will, what the outcome of that will be. And also it's done by surgeons. We are cardiologists, we don't want to give work to surgeons, okay? So we're developing an in situ injectable tissue engineering a process. The idea is that you will inject the polymer in a liquid state into the infarct together with the cell, and then induce polymerization and generation of the tissue in the heart. The polymer will serve a number of processes. For example, it will prevent the cells from being lost acutely. It will entrap the cells in the area, provide a protective environment, and also it can replace the missing extracellular matrix and impact remodeling. It could thicken and strengthen the scar, prevent the expansion of the scar and thinning of the scar that results in increased load and dilatation of the ventricle and continuous deterioration. So it might affect remodeling just by the mechanical properties of the polymer. So together with Dror Selectara, the biomedical engineering, he generated hydrogel, which is based on PEG fibrinogen. You could change the properties of this PEG fibrinogen by adding more PEG or less. You change the stiffness, the degradation process, and so on. So we inject it into the infarct. You can then polymerize it by a number of ways. In this case, we use the photoinitiator and UV light, but you can do it as a temperature dependent and so on. This is how it looks in histology after 7 days. After a month, most of it will be degradable. Here is with cells, and it seems like the animals like it. These are two control. These are the two groups that we studied. You can see that this is an infarct in which cell line was injected. This is how it looked 2 months after creation of the infarct. You can see a very thin infarct with dilatation of the ventricle. In the group that received both cells and hydrogel, you can see how thick the anterior wall is. There's no thinning and there's no dilatation. So when we compare the 4 groups, here we show only fractional shortening. Remember, it starts about 40%. It goes down after the infarct to about 20%. And you can see that in the cell line group, all animals deteriorate. Note that just by injecting polymer into the remodeling infarct, this alters remodeling, so there's no deterioration. Cells do a bit better than the polymer, but not significantly, and the best results we get when we have cells and the polymer. Okay, so Optimal regeneration would require you to regenerate the extracellular matrix, the cardiomyocyte, and probably also vascular cells. That's what we are learning. One of the problems in using embryonic stem cells is that you cannot generate them from an adult individual. You had to go all the way to the early stage blastocysts to get cells that are pluripotent, can become any cell type. This limits you use in regenerative medicine because as we are dealing with a non-autologous transplantation, not from the same patient, and as well it limits your use of these cells for modeling the patient-specific heart tissue. Now these challenges may be solved by the technology introduced by Shinya Yamanaka, for which he was awarded the Nobel Prize last year, in which he took somatic cells, adult cells, such as fibroblasts, used the expression of 4 transcription factors in these cells to reprogram these cells to become embryonic stem cell-like cells called induced pluripotent stem cells. So you take an adult cell and you change its phenotype back to a state resembling the fertilized egg. You then take these induced pluripotent stem cells and differentiate them to any cell type that you want, and these cells should have the patient-specific genetic properties. So this turned out to be a really robust technology. We can now routinely take fibroblasts from these patients, as you can see here, reprogram them using Yamanaka technique, coax the differentiation into the cardiac lineage using your favorite differentiation method, and we can start with skin cells and end up with the patient-specific heart cells beating in the dish. These cells have cardiac-specific properties. You can see the development of sarcomers. One can record action potential from these cells. You can load these cells with calcium-sensitive dyes and get intracellular calcium transient. You can show that they have intracellular calcium stores by giving caffeine, for example, and so on. Similar to the human ES cells, you can plate them on the multi-electrode array plate and get that these cells display a functional syncytium and so on. The next question we ask, can we generate these cells also for the patient that will eventually need them? So we attempted to create these iPS cells from patients with heart failure. So patients that had large myocardial infarction, that have advanced heart failure, that have sick hearts, multiple comorbidities, that are elderly, and whether we can get iPS cells and have healthy heart cells from them was a question. We didn't know that. So we compared the ability to generate these heart failure, these iPS from patients with acquired heart failure versus the ones that generated from foreskin fibroblasts. These are, 7-day-old fibroblasts. So this is the earliest human fibroblasts that we can find. And you can see that in all cases we can generate induced pluripotent stem cells. These are the cells stained for pluripotent markers, OCT4 and NANOG. And we can differentiate them to generate cardiomyocytes. As we will talk later, these cardiomyocytes are early stage, healthy, unlike the patients from which they were generated and which have severely dysfunctional cardiomyocytes because of his heart failure. So it's possible to take a heart failure patient and have cells that are similar to the cells that they was born. These cells again can integrate with existing heart tissue in the dish and generate a functional single syncytium. We can transplant them and they will survive, generate gap junctions with host tissue, and so on. Okay, so more and more people are thinking of using these cells for other applications beyond regenerative medicine. We'll talk then about the potential for using them for drug development as for modeling cardiac disease. And we talk about also the challenges about using these cells. So one potential application of these cells is for studying, for example, drug interaction in the heart. You can look at drug screening for adverse effect, and the most common effect that is being looked on by drug companies is a potential to screen these cells for hERG, blockade, and QT screening. The idea is that many drugs, up to 50% of all molecules being developed, have kind of, as you all know, effect on blocking the hERG encoded channel and may prolong action potential duration and give rise to early after depolarization and proarrhythmia. So we can show that these cells have a hERG current and that they respond to hERG blocker by increasing action potential duration and giving rise to early after depolarization. We can test different drugs on their effect of them, or look at their drug response properties, concentration properties, and so on. We can do it at the intercellular level, but also at the extracellular level. So when we measure something which is similar to a QT interval by extracellular electrograms, called field potential duration, you can show that you can give different drugs that will affect QT interval, either increase or shorten it, and you can identify it in a dish. Interestingly enough, this is a study that was recently published from Stanford showing that the background, the genetic background of the patient may have an effect on its response to drugs, okay? So, of course, not all people respond the same to drugs. Some people are more sensitive to it. For example, in this case, he took heart cells from different type of patients, from healthy patients, from patients that had the congenital long QT syndrome, patients with hypertrophic cardiomyopathy, and people with dilated cardiomyopathy due to genetic disorders. And he looked at the response to this cell, I think in this case was cisapride, which blocks IKR, and you can see that different cell types with different genetic backgrounds acted differently. All of them increased action potential duration. Some, it happened at only high doses, as you can see here. Some, it happened at low doses with a significant arrhythmogenic behavior, earlier the polarization triggered beats. So one could imagine that you can have a repertoire of different iPS cells with different genetic backgrounds. You could take cells that responded with arrhythmia to drugs versus one that do not, and that you can try to identify screen drugs on different group of patients. Okay, and the second application of these iPS technologies that I would like to discuss with you is the potential of using these cells for modeling inherited disorder. The idea is that if you have a patient that has a cardiomyopathy or inherited arrhythmogenic disorder, you can take cells from this patient. Since you can study directly the patient's heart, at least until what I learned today, you can take the skin cells, reprogram them to generate iPS cells, make heart cells, and these heart cells should display the patient-specific properties of that specific patient. One can then study these cells with a variety of techniques and to try to study the impact of the genetic change on the phenotype of the cells, with the hypothesis being that the cardiomyocytes should display the patient-specific disease. So we have a great interest in familial arrhythmogenic syndromes, both from a mechanistic point of view and also because these are the patients that we treat. These are also very good disease states to start with, because we would like to start with diseases that are cell autonomous, meaning that you can identify them at a single cell level or the tissue level. You don't need an entire organ or entire organism, because of course we cannot simulate it in the dish. They are monogenic with a relatively clear genetic phenotype interaction, so we would like to start with this type of diseases, not with polygenic complex phenotypes. They start at a relatively young age, and what I told you until now is that these cells are relatively early stage in terms of development. So this is also important. For example, although there are now studies of that, if you think of Alzheimer's that develops at the age of 80, these cells might not be a good model for that, although there's a caveat to that. We'll talk about it. So these inherited arrhythmogenic syndromes can be grouped into those that affect ion channels, such as the long QT syndrome, short QT syndrome, Brugada syndrome. Those that affect calcium handling, such as the case of catecholaminergic polymorphic ventricular tachycardia, or CPVT, and those that affect cell-to-cell contact, such in the case of arrhythmogenic right ventricular cardiomyopathy. So we started with the long QT syndrome, and this was the first study that demonstrated the ability to model a functional disease using the iPS in the heart. So what we did is, as you all know, the long QT syndrome is due to mutation in ion channel. most of them due to loss of function mutation in potassium channeling, impacting repolarization, resulting in delayed repolarization, prolonged action potential duration, manifested in the electrocardiogram as a prolonged QT interval, and this prolongation of the action potential duration will give rise to early after depolarization and to this malignant arrhythmia called torsade de poids. So a few years ago, we had a young female patient coming into our department after she had cardiac arrest in the street. She was resuscitated and brought on to our cardiology department, where she had multiple episodes of this VT, what we call VT storms. We implanted a defibrillator in that patient, and we learned that several members of the family died at a young age, and we noticed that she had a prolonged QT interval, as you can see here. We screened the family and we identified a point mutation in the KCNH2 gene responsible for the long QT type 2. Lucky for us, not so much for the family, this was a very malignant mutation affecting the pore-forming region of the channel. So we took this opportunity of taking skill cells from these patients, reprogramming them to generate iPS cells, and then coaxing the differentiation of the iPS cells into heart cells and then studying them. So you could see that when we record action potentials from control cells, you can record action potentials that resemble three types of cardiomyocytes: those that resemble more ventricular cells, those that resemble more atrial cells, and those that resemble more nodal cells or pacemaker cells. In the long QT cells, you could see prolongation of the action potential duration both in ventricular cells, in the atrial cells, both during spontaneous beating as well as fixed pacing. You can see this at the single cell level, but also at the tissue level. So when we record from a cluster of cells, you can record something that we call the field potential duration, which is analogous to the QT interval, which is significantly prolonged in the long QT cells. But you can also see it in your eyes. When you look at the contraction of the long QT cells, you can see that it's prolonged in the cells. Note the contraction and the long time until it takes for relaxation. So when you look at that in the dish, in contrast to the healthy control cardiomyocytes, which have a shorter contraction time, as you can see here. Importantly, these cells displayed early after depolarization, as you can see in this example. And some of them also developed triggered beats, as you can see here, both at the single cell level. You can also identify it on the multicellular level. Now one can test the effects of drugs on these cells. Here, for example, we try to shorten the action potential duration by giving pinacidil, which is a K-ATP channel opener. And you can see that by opening the alternative potassium channel, this shortens the action potential duration and prevents these EADs. So you can string the effects of drugs, new or existing drugs, on these cardiomyocytes. The second type of disease that I would like to discuss is called catecholaminergic polymorphic ventricular tachycardia, or CPVT. CPVT is, again, an inherited arrhythmogenic disorder that may result in death in young people. There are two types. The most common is the dominant inheritance form, called CPVT type 1, which is due to mutations of the ryanodine receptor, okay? Resulting in a leaky ryanodine receptor during diastole that results in leak of calcium out of the SR during the diastolic period. This activates the membrane sodium-calcium exchanger that leads to depolarization of the membrane, what we call delayed after depolarization of DADs, that may give rise to this biventricular polymorphic VT, typical of these CPVT patients, which is really typical during emotional or exercise, emotional stress, or during exercise, during adrenergic stimulation. The less common type is CPVT type 2. Interestingly, it was first reported in Israel in a family that we are taking for in the hospital by Michael Eldar, in which you can take, this is a recessive disorder resulting in mutation in the main buffering, calcium buffering protein in the SR called calsequestrin. Again, this leads to calcium leaking in the diastole and the same phenomenon. So we studied a patient's a family with CPVT type 1 together with Shiva Hospital. We took, this is the index patient. You can see during stress test, the generation of polymorphic VT. We can take cells from these patients, reprogram them to generate iPS cells, then differentiate them to generate cardiomyocytes. This CPVT is due to a mutation in the ryanodine receptor gene in this family. We can then study these patients, and this is control cardiomyocytes, as you can see here. The CPVT cardiomyocytes, to a great extent in many cells, develop these delayed F33 polarization, mainly during phase 4. Occasionally we see during late phase 3 in these cardiomyocytes. And they were really sensitive to adrenergic stimulation. If we give them forskolin, which increases cyclic AMP, or give them isoproterenol, you can see that at baseline, you can see these DADs between the action potentials. Activating the adrenergic pathway significantly increases the number and amplitude of these DADs and also results in triggered beats, as you can see here. Another way to show this is to pace the cells fast and then to stop. You can see the DADs at the post-pacing interval, which significantly increase in number and amplitude and duration and leads to triggered beat in these cells. Now, there are some the major treatments for CPVT is beta blockers, okay, to prevent this adrenergic stimulation. But for patients that fail this disease, the recent option in the field, the new kid in the block, is flecainide, and it was shown that flecainide may reduce arrhythmogenic event in these patients. The mechanism is under debate, whether it is a stabilizer of the ryanodine receptor or whether it just prevents excitability but blocked by sodium channel and blocking the vein. In any case, we saw that flecainide prevented the DADs in our cells. Since the problem again is calcium handling, we went on to do calcium imaging in these cells. This is a control cardiomyocyte. You can see that they have calcium transient. In some cells, we can see these local calcium release events or spark events in these cells. In the CPVT cardiomyocyte, we saw gross abnormalities. We could see a lot of sparks. Some of them are large and wide local calcium release events. We could see this double humped potential calcium transit in this cardiomyocyte, and occasionally these large release events in these cells. So there was significant calcium handling abnormalities.
Lior?
Yeah.
I'm curious why you're calling them sparks and not just
spark-like or
transients that underlie DADs or...
we didn't do simultaneous action potential recordings to see what they give rise to in terms of electric.
I mean, sparks are kind of what are the elementary events.
Yeah, but when you look at them, you can see spark-looking events. And these are maybe larger sparks that are grouped together or something like this. We don't call them sparks, we call them local calcium release events, okay? To stay away from this train. The next thing we wanted to see is whether we can recapitulate something which is called SOICR, or store overload induced calcium release. The idea is that the normal way calcium is released from the SR is through a calcium-induced calcium release mechanism, in which calcium that enters to the L-type calcium channel binds to the ryanodine receptor and results in release of calcium. There's an alternative mechanism with a putative luminal receptor in the SR, in which if the free calcium within the SR increases a certain threshold, it can activate this theoretical sensor and lead to spontaneous calcium release not linked to an action potential. And the thought is that maybe a ryanodine receptor mutation lowered the threshold for this SOICR event. So to test this, we tested these cardiomyocytes by increasing extracellular calcium concentration, as you can see here. In the control cardiomyocyte, you can see that the physiological calcium level, we chose quiescent cardiomyocytes. There were no spontaneous release events, but when we increase calcium levels to a very high concentration, you can start to see these SOICR events. In contrast, in the CPVT cardiomyocyte, you can see these SOICR events also at very low level of calcium concentration. You can see the differences in this plot summarizing. So it seems like The threshold for SOICR, at least using extracellular concentration of calcium as an indicator, was reduced due to the ranitidine receptor mutation. We are also starting to study the second type of CPVT. As I told you, it's a recessive disorder, which is really malignant. We have very severe disease phenotype in these patients, in these kids. There were many number of deaths. Even when we implant defibrillator in these patients, it's not doing so well because they get a shock, they get VT, they get a shock. The shock induces a pain and it results in a burst of adrenaline, which generates another arrhythmia, which gets another shock, so they come in occasionally with 30 or 40 consecutive shocks and they have a miserable life. As you can see, these cells have really abnormal calcium. Here you can see a train of probably DADs, as you mentioned. So there's a lot of, in the CPVT mutations, a lot of ectopic activity, both in calcium, and I didn't show the intracellular recordings. And we again see that they have abnormal SOICR. In this case, it's probably not due to, or maybe it is, we didn't look at the mechanism, to a reduced threshold of the ranitidine receptor. Maybe it just increased free calcium because of the calcium question. So the mechanism has to be discovered more. So in both cases of CPVT, we could see the abnormal phenotype in this cardiomyopathy. And the third and last disease that I would like to discuss today is a disease called arrhythmogenic right ventricular cardiomyopathy, or ARVC. Yes?
Have you ruled out a difficulty in differentiation of the cells with the different mutations? Because if they're at a different stage of embryonic development, they rely more or less on calcium.
Okay.
Like, for example, neonatal cells with more calcium.
So this is a very good question. So the way the field has done it until now is to study a number of patients with a mutation, a number of clones versus a number of controls. And if you see a big difference between the two groups, then you could say it's probably not a matter of differentiation because the control cells will differentiate in the same manner, in the same stage, and so on. More and more we are moving, and probably this will become the standard of practice in the field, is to correct the mutation. Okay, so now with gene editing, CRISPR, TALEN technology, it's possible to change a single nucleotide in the DNA wherever you want. So you can take an isogenic line from the same patient, create a mutation, or correct the mutation. So if you can correct the mutation and rescue the phenotype, this is a very good mechanism to show that indeed the mutation is responsible for the phenotype.
But the cells look otherwise the same?
The cells control and CPVT, yeah, they look the same. Pretty much the morphology looks the same. We looked at the electron microscopy, we looked at ranitidine receptor staining. Seems the same amount and distribution of the ranitidine receptor in the control cell. In general, when we talk about this limitation, one of the limitations of this technology is that these cells are relatively immature. They're not really adult heart cells yet. They have a lot of features that correlate with adult heart cells, but are not there yet. And this is one of the challenges, how to make them more mature to resemble more the adult cells promoted.
These cells have, for example, a T-tubule?
So T-tubule is a problem. I think part of it is because you culture them as two-dimensional and they don't have a lot of Z, in the Z-axis, so they don't have developed a T-tubule. So this is a big problem. They resemble, I don't know if it's a big problem, they resemble more atrial cells or in this regard of T-tubules, if you look at calcium handling. So one of the challenges.
I'm sorry, what about your calamari cells?
With the data, that's the next step. We want to use this tissue engineering in which we think that there is maturation to use that for modeling. The problem is that until recently, we didn't have good differentiation protocols. So for this calamari cell, you need a million cells. So it takes much more effort to generate
the three-dimensional aspect.
Yeah.
T2.
Yeah. I think we starting to look at it. We didn't have create enough of them to look at that. Now that we have protocols that we can generate many cells, we will go back and try to do all our models using this technique, both to look what happens at the tissue level, as well as to disperse the cells to see whether they are more mature using this technique. Okay, well, so that's the next step. But if I go back to the third disease, and then we can discuss this more. So this disease called arrhythmogenic right ventricular dysplasia is a very interesting disease. If you look at pathology, what you see is replacement of heart cells in the right ventricle primarily by fatty tissue or fibrofatty tissue. And interestingly enough, many of the patients in the last decades were identified to have mutations. So this is a genetic disease. And the mutations are mainly in proteins that are involved in desmosomes. So they are desmosomal protein mutations. So interesting how a structure that is involved in cell-to-cell contact results in this phenotype and primarily in the right ventricle. Okay? Anyway, this results in arrhythmias. There are two stages of arrhythmias. One in this advanced stage where you have this type of morphology where it's probably mainly just reentry. And one in the early stage before you have this advanced replacement of heart cells with this fat infiltration, where they have limited structural abnormalities, which is also interesting. Why do you have this first phase of arrhythmias? I think there are some results in the literature and what I'll show you that may explain this. So we studied a few patients with ARVC. This is an interesting patient of mine. He's a Ph.D. student at Technion that is into extreme sports. He does these ultramarathons and things like this. And during while he was running or something, he came with a very fast VT. It's about 180 beats per minute. He had it for hours. It didn't stop him from running. And he came to the ER. And we had learned that he has a lot of these. We mapped his ventricle. This is a study that we did many years ago showing that you can identify the ARVD diseased areas by low amplitude electrograms. So the colors here represents the amplitude of the recording electrograms, and you can see that we can have most of his right ventricle had low electrograms, meaning that there was no cardiomyocytes there. We identified a mutation in the main desmosomal protein, plakophilin 2. This was a rather novel mutation that results in an early stop codon and lack of this protein from this allele. So we created iPS cells from this patient and another patient, and then started to study them. So the first thing we obviously looked was staining for plakophilin 2, the mutated channel, and we can see that this is staining for plakophilin, the green point in the healthy control cells in this cell here, and we can see reduced staining for plakophilin in the ARVC cardiomyocytes versus when we quantify it in many cells, we can see significant reduction in the plakophilin immunosignal in the diseased cells. This is not surprising. What was a bit surprising, although when you scan the literature, this was also shown, is that we have reduction in the immunostaining of an additional protein in the desmosome, plakoglobin. So when you have plakophilin mutation, it affects desmosomal structure and results in reduction of staining for plakoglobin in the membrane. And we also noted that it can affect neighboring intercalated disc structures. For example, we see reductions in connectin 43 staining, and also there are some hints that they also may affect the sodium channel. And this is consistent to what Mario Del Mar shows in his model. So the mutation in desmosomes may affect other intercalated discs, and may explain, for example, why we have an early events of arrhythmias, even we don't have still this fibrofatty infiltration, just by effect on connexins and the sodium channel that may result in slow conduction. But what was interesting is that we looked at electron microscopy. So this is how this desmosome looks in these early iPS cells, derived cardiomyocytes. And when you look at the ARVC cardiomyocytes, not in all cells, but in many of them, we see abnormal desmosomal looking structures. You can see the hazy appearance and the widened external and internal gaps of these cells, which results in significant differences in, for example, dimension of the desmosomes. So mutation in the plakophilin results in a structurally looking abnormal desmosomes. Again, not in all cells. And what was striking for us, what was interesting, is that in some of the cells we can identify the presence of lipid droplets. So this is electron microscopy of one cell. You can see lipid droplets accumulating within the cell, the nucleus, the cytoplasm, the lipid droplets. And you can see the abnormal desmosome in this cell. This is the membrane. And when you correlate the presence of abnormal desmosomes and the chance of seeing this, we can see that this is really cell autonomous. So cells that had displayed normal desmosomes, for example, here characterized by normal width of the desmosomes, did not have this lipid droplets accumulation. While cells that had the highest abnormal desmosomes had a good chance of accumulating lipid droplets. So it seems like this could be a cell autonomous phenomenon, at least in these cells, which abnormal desmosome leads to accumulation of lipid droplets. And that you can make this phenomenon severe by exposing the cells to lipogenic stimuli. So this is how the cell looks after we add a medium with fatty acids, insulin, and something that increases contraction, increases cyclic AMP. So you can see that it almost looks like an adipocyte. So there's complete change of the phenotype of the cells from a cardiomyocyte to an adipocyte-like structure, and we also see expression of the pro-adipogenic mastogene PPAR gamma in these cells. So what's the mechanism of this? So this is a bit provocative because, like I told you, most people believe that the cardiomyocytes die and are replaced by fibro fatty. We say that some time potentially leading to this pathogenesis is transformation of cardiomyocytes to adipocytes. Whether this is in the progenitor cells or what type of development, we don't know. What's the mechanism of that? So there's a few hypotheses. There's one hypothesis that says that plakoglobin, which is the other protein in the desmosomes, that due to plakophilin, the abnormal desmosomal structure, is released from the desmosome and is released to the cytoplasm into the nucleus. And plakoglobin is also called gamma-catenin, which is very similar to beta-catenin. So it may interact with an important intracellular signal pathway called Wnt pathway, and may alter gene activity and may result in this phenotype by altering Wnt pathway. And we have data showing that indeed there is reduced Wnt activity in these cells by looking at the nuclear density of beta-catenin in the nucleus. So we thought if indeed this is the mechanism in which Wnt activity is altered in these cells, leading to this phenomenon, if we rescue Wnt activity, for example, by giving a small molecule called bio, then maybe this will prevent this to happen. And indeed, if we give bio together with this lipogenic stimuli, this is how the cell without bio looks, develop this adipocyte-looking structure, while the cells that receive bio, you can see this prevents this phenomenon. So this could be a potential mechanism by how this disease developed, and potentially could maybe prevent this in the future. So I'd like to summarize and tell you what we think iPS cells can allow us before discussing some of the limitations. So we think it could be very interesting for personalized medicine, for drug development, and for regenerative medicine. The idea is that you can get patient-specific heart cells that will allow you, for example, to screen safety and toxicity screening, looking at effects of drugs on QT and contractility, which are the main two problems of drugs. You can look at in a patient-specific manner on a group of patients with a specific genetic background. You can potentially maybe even individualize drug therapy, patient-specific phenotype of a cardiomyocytes. You can try to see what is the best drug to give to that patient. What I think is would be very important, you can generate disease cardiomyocytes like ARVC, long QT, CPVT, dilated cardiomyopathy, and use this as a method to develop new drugs. You can think of it as a new unbiased way to screen big libraries of molecules to try to see which could rescue phenotypes, and then start looking for new targets in these cells, because you have now human disease cardiomyocytes. We talked about regenerative medicine, that it could be done in a patient-specific manner. And as we discussed now with gene editing, CRISPR and TALEN technologies, you can correct mutations. So perhaps you can have a patient with genetic disorder, have iPS, correct this mutation, and generate heart cells that will be normal. Okay? That can, heart cells or other cell types, that could be fixed in terms of genetic disorder. So what are the problems, at least in the disease modeling and drug disease? First of all, there's the issue of incomplete reprogramming in epigenetic memory. This is becoming more and more solved, the issue of generating iPS that are not true iPS. Regarding cardiomyocytes, we still have a big issue of heterogeneity and immaturity. So these are the two major challenges. First of all, after solving the issue of making enough heart cells, the next question is how you can make cardiac-specific cardiomyocytes. So can you find signaling pathways to generate only ventricular cells, or only atrial cells, or only Purkinje cells, or only pacemaker cells, and so on. And there are studies now starting to show that there are indeed these pathways that you can generate. For example, there was nice studies that can make pacemaker cells, or atrial cells, and so on. So this is the next frontier, making subtypes of cardiomyocytes. How to mature them, because we talked about the problem of early stage in terms of electrophysiological phenotype, calcium handling, morphology, and so on, metabolism. So this is a big challenge. We need to move to see whether we can move from modeling the diseases that I described to you as severe phenotypes. So, you could easily see it. Can you see diseases that have less of a severe phenotype? Can you model penetrance, issues relating penetrance? Can you look at polygenic disorders? Okay, can you look more subtle differences? So you need to solve this problem in order to go forward. And as we talked, we are looking on phenomena that are on the single cell level. Can you make it a more clinically realistic model? Can you make an engineered tissue that will have multiple cell types to try to recapitulate the heart, the adult heart, to make it a better model for screening and so on? And so we have a lot of work to do, and that's good, because we have what to do. So with that, I'd like to end and thank the people that did this work in my lab. Mainly the earlier work was Tachycardia. He was the first graduate student in my lab. He's very talented. Now he has his own lab at the Faculty of Medicine. Most of the modeling work was done by Oren Kaspi, Lanit Yitzhaki, and Leonid Meisels. The iPS work was done by Lior and Oren. The tissue engineering work was done by Oren, Manal, and these are collaborating people in Haifa. This is a typical house in Haifa. This is how we live in Haifa. So each PhD student gets this type of house. Collaboration. These are collaboration in the world, and I'd like to thank you for your attention. If there's more questions, I'll be happy to.
Thank you, Lior, for a fantastic talk. Any questions or comments?
Yeah.
On your biologic pacemaker slide, you made the comment that you selected for spontaneous heart cells.
Spontaneous beating.
Pardon?
Spontaneous beating.
Spontaneous beating. Then you mentioned the subtypes later on here at the end. So did you have techniques to select for the sinus node or atrial or ventricular?
No, at the time, no. We just took clusters of about 1,000 beating cells. We hypothesized that within them we had pacemaker cells that allowed them to beat. And we transplanted many of these clusters into the heart. The idea is that we had pacemakers within the cluster, they will start pacing, the action potential will propagate within these clusters, and that will have many interaction with the ventricular cells that will break into the peak heart. That was the idea behind it. And we only looked at it for short periods. So whether these cells, pacemaker cells, become ventricular cells after transplantation and stop pacing, whether they are rejected, we don't know. We just focused on a short period. And most of our efforts, because of the magnitude of the problem, and we have good solution because pacemakers work quite well, we're investing in the heart failure.
Do you have any sense that the cells do or might remodel once they're in situ? So is there any validity to perhaps putting things that aren't completely differentiated into some form in there, and then they might get differentiated further by the environment they're in and the need for what they're doing?
Yeah, so this is what we hope for. And in fact, if you differentiate too much ex vivo and transplant them, they don't survive well. Okay, so there's a conflict. If you make them more mature and adult, they don't survive on transplantation. Yeah. So our hope is perhaps that they will continue to mature with time. We're now spending efforts creating lines that will have fluorescent sensor of calcium and voltage that we can follow them over time. There's been some studies, mainly using mouse embryonic, a little bit in human, in which people are creating slices from the transplanted cells and showing that with time there's maturation of the active potential properties. Not to a completely adult, but significant maturation in terms of the action potential properties. Probably the environment will help that. Yes.
At what age are you actually doing the transplant? At how many days?
About 30 days after differentiation. 30 days of differentiation, about. Yes.
So just a naive question. I have seen like a couple of talks on STEM, iPSCs, and everybody talks about the heterogeneity of iPSC. But when I look at the data, they look at so certain channel properties, action potential properties. Actually the error bar is really small. So where does that heterogeneity come from, and how is this?
The error bar was we classed them to different types. Okay, atrial, ventricular, and nodal. So each one was it has its own action potential duration, its own. Now.
But so if you say heterogeneity, there must be something in between, right?
Between these three,
yeah.
In each of these clusters, you see differences in terms of action
the
potential.
differences are like just so say something or something.
Different,
like that.
100 milliseconds, 200 milliseconds, because they're very, very long. I don't know what the difference is. Maybe you guys can tell what's the differences if you see from human cardiomyocytes that you disperse from hearts. What's the differences in the action potential, the dispersion that you see?
Dispersion in a normal heart.
No, not in the intact heart. When you disperse them into single cells.
You have to go to him. He's the only one doing that.
Something with action potential. I'd be quoting the literature.
So what's the literature?
I think Jamin's point was that even though you have this heterogeneity, i.e. you have different types of cells, what he's saying is that within each group you're saying that there is very little difference. So does that mean that you have cells that are here, and here?
Yeah.
Otherwise, how did you get such tight groupings, is what he's saying.
So how did we group between the atrial and ventricular and well, you take like parameters like APD 90 versus APD 20 to differentiate between triangular shapes and the more of a plateau, which we call the ventricular line.
But then his point is, once you do that,
then,
there apparently is no
that's
difference.
not that bad.
Okay, that's the point.
It's not that bad.
So they do fall into clearly distinct clusters, is the point.
Yeah.
I see.
Is there
So,
any way-
but you don't know how they fall into different groups.
No. So one of the things we want to do is to try to correlate, if we're able to set it up, to do a single cell transcriptome analysis from the cells that you patch, whether they are indeed, atrial cells, ventricular cells, there is a correlation with this. Yeah. Yes, you.
Can we look at connexin expression as a marker of different mindset?
So again, within the EBs initially, we looked at both, they expressed both connexin 43 and 45, not 40, interestingly. We didn't have a good antibody for that, no. But they express connexin 43 and 45.
She does.
A good antibody?
Connexin 40.
So the antibody we had, I don't know, maybe it's probably, we didn't see connexin 40 stain.
So you mentioned Mario Delmar's work about sodium channel subunit distribution is related to the connexin. So if that work is correct, then you would expect your cells have a much lower density of sodium.
Mm-hmm.
Is that what you find?
So we haven't did the ARVC. We haven't done the single cell analysis yet, but we looked at multi-electrode array data, at extracellular potential, and they have reduced upstroke in extracellular, and they have lowered the conduction velocity in these maps, hinting that there might be also problems in the sodium channel over the connexin as we saw. But we need to do the single cell recordings to look at the sodium curve. Yeah.
In your ARVC model, did you have to give a lipogenic stimulus to get the fat? Or did it?
No.
It happened after?
You could see that it's not as prominent, but if you look closely at electron microscopy, the ones that have desmosomal abnormalities usually have these, about half of them have these lipid clusters within them. But you have to look for it. I imagine that if we wouldn't have done electron microscopy, we might have missed it if you look at other ways of staining lipids. So it was very evident by electron microscopy. And there has been 2 other papers that were published that show similar results, which makes me think that it's, we were a bit hesitant at the first, because it's like the pathologists always say, we don't see this, where does it come from? But what I think is that they never look at such at the early stage, that this is like a developmental issue.
With this ARVC, do you see fractionated electrograms or only?
We need to look closer again to see how the signals look like. Again, we're now moving more and more. We're setting it up again to generate monolayers. Since we now have this monolayer differentiation that gives you 100% cardiomyocyte, we want to put this monolayer on the multi-electron.
It's interesting to see because if there is some fibrosis there or
Mhm.
replacement by fatty tissue,
And then it might be.
then maybe there should be some
Mhm.
discontinuous conduction that could be actually expressed before you can see anything on MRI.
Mm-hmm.
And
Yeah.
it could be a very important.
Mm-hmm. I know. Tell the truth, initially I thought that when Oren started this project, I said there's no way that we'll identify anything because this is much complex disease to see at a single cell, level or cell phenotype. It probably require a lot of interactions and inflammation and things that you can only see in the but it was really, really robust. We see it in a number of patients.
Any other questions, comments? If not, then happy Thanksgiving, everyone.